Good morning, everyone. Today we're experimenting a little bit with some live streaming through YouTube that won't impact this room, but for those who might not be here who are connecting, please let us know if you encounter any problems. That's only for the Broad before we go public with it. Today we're very lucky to have Geraldine van der Auwera, who will be talking about sequencing and variant discovery pipelines. Geraldine is the manager for outreach and communications for the Broad Data Sciences and Data Engineering group, and we're very fortunate to have her speaking. Thanks very much.
Thank you very much, Chris. All right, thank you everyone for coming up, showing up very early this morning. I really appreciate it. Or for the people who are watching us on YouTube from the future, if you can let us know how the election turns out. I just can't take any more of the current campaign, so just let us know. In the meantime, we're going to talk about something more uplifting. It's the amount of progress we've been able to do in the to make in the field of sequencing and variant discovery. We've actually got a lot of change relative to last year when we did this, essentially a primer on the same topic for this primer series. But a lot has changed, and so I'm quite excited to tell you about all that's been happening this year. I'm going to talk about sequencing and variant discovery, of course. But first, I want to acknowledge the fact that this is the work of a lot of people. There's a very large team, multiple large teams that are working on this. I couldn't begin to list the names of everybody that's involved. This picture just gives you an inkling of everybody that's involved. It's a really wonderful synergy between the Genomics platform, which many of you know as the sequencing facility at 320 Charles, as well as the data sciences platform, which is a fairly new thing. We only got a name this year, but I think we've existed for about 2 years. Data sciences and data engineering group, which I'm part of, is part of that umbrella. And our job is to develop the methods, the software, and the infrastructure to do the analysis of the sequence, especially with regard to variant discovery. So that's what I'm going to focus on today. Oops. Yeah. Okay, so the scope of this presentation is going to cover 3 main things. I've broken it down in these 3 topics. The science is really what the data looks like, what transformations need to be applied to the data, and how we formalize the set of recommendations, the set of things that need to happen in what we call the best practices workflows. So that's going to be the bulk of my presentation. Then I'm going to talk about the software that's involved, just to give you an easy reference to look up what software packages, where to get help, where to get more information about what they do, fairly briefly. And then I'm going to talk about the concrete pipelines, the actual concrete implementations of those idealized workflows, and tell you a little bit about how they're run at Broad and what has been changing and evolving. Okay? So let's start with the science. So I'm going to talk about the science of what happens in the variant discovery pipelines. The ultimate goal here, I think we'll all agree, is to identify genomic variation in sequencing data, specifically high-throughput sequencing. And you'll notice that I say high-throughput sequencing and not next-generation sequencing because, let's face it, it was next-generation sequencing 10 years ago when I was in grad school. Now it's just sequencing. So I call it high-throughput sequencing or sequencing in general. So we're looking for, we'll have a large file of sequence, and we want to use that to identify genetic variation in individuals relative to a reference genome. That's the main thing. At this point, we distinguish germline variation, which we inherit from our parents, from the somatic variation that arises during the development and life of the individual, which can be benign in many cases, but can also lead to cancers. And some of the algorithms that we'll need to apply are going to be different based on what we're interested in identifying. We do all this, as I mentioned, relative to a reference genome, which is a standardized genomic sequence that is meant to represent a wider population of the organism. In the case of the human genome reference sequence, there has been an iterative refinement of what we use as a genome reference. Initially, efforts to represent what is an average human with a single sequence started out kind of primitive. It's been refined over the years. The latest version is called HG38 for Human Genome 38, or GRCH 38 for the Genome Research Consortium version. And this is kind of an exciting development in the sense that it adds a lot of nuance to how we represent human genomes. It adds the ability to represent alternate haplotypes that correspond to populations that diverge significantly in certain regions of the genome from our original standardized reference. And that's important and to be able to capture variation in populations of humans that were divergent from that original version of the reference. Okay. For other organisms, so all the principles that I'm going to talk about today apply equally to other organisms, non-humans, as long as you have an assembled genome available. If you don't have that, somebody's out of luck. But you can always create that assembly. But everything we're going to talk about is relative to that reference, because that way, when we accumulate data from multiple individuals, we have a common reference to compare them. Okay? Now, there's different types of variants that we're going to be looking for. The simplest ones, the short variants. Here, I'm representing at the top the reference sequence. And here, each bar here is an individual sequence read. And you see here that if I look at the pileup of reads in this region, this person has a point mutation, which we'll call a SNP or SNV for single nucleotide polymorphism or variation. And we also have an indel here. So the SNP is where you have just a single nucleotide change. The indel is when you have an insertion or deletion of sequence. We generally consider that indels can go up to a few hundred bases. Beyond that, we're going to consider them as a copy number deletion or structural variation. Copy number variation is when you have an amplification or deletion of a region of the genome, for example, through a chromosomal duplication, various other events. Structural variation is when you have a translocation or change of orientation of a particular genome region. And these are going to tend to be larger events than SNPs and indels. And so these are all different types of variants. When we're looking to identify these in real data, we'll use different approaches and algorithms. I'll talk a bit about that later in my talk. Okay, so another thing to consider is that there's different types of experimental designs that are available to us for this task. The main two that we distinguish are the whole genome sequencing and the exome. The exome, as well as things like gene panels, which are all targeted ways of sequencing data. In the whole genome, as the name implies, we're capturing sequence from across the entire genome, with the caveat that there are some dark regions of the genome that we can't quite access with the current state of the technology. These are shrinking, but they're still there. So when we say whole genome, it comes with a little asterisk. But generally speaking, we're capturing all of the sequenceable genome, and we're going to have fairly even coverage so amount of reads at a particular site, across the genome, and we'll be able to discover variants anywhere, whether they're in intergenic regions, whether they're within introns or exons of genes. In contrast with targeted sequencing experiments, there are some restrictions, limitations, because, for example, in the case of the exome, this is a subset of the genome that we've targeted specifically. Typically, that is targeting a set of exons from identified annotated genes. And we're only going to have sequences in kind of piles of sequences around exons. Now, there are several complications with that. One is that the profile of coverage is going to be very uneven. Instead of having a nice big blanket of reads across the genome, here we're going to have piles of reads that are localized, which from a technical point of view is going to add some noise to the models. It's a little harder to deal with that technically. There's also the fact that there are some regions where you won't have coverage by design, and as a result, your power to discover variants is limited to what is targeted in the particular kit of the manufacturer that you decided to use for your experiment. So it's important to keep in mind that when you're selecting an exome preparation kit for your experiments, you should check, well, what other studies am I going to want to compare my results against? What kits are my collaborators already using? You want to use as much as possible the same targets and so on as your collaborators. Because otherwise you get batch effects, things are less comparable. So overall, exome comes with a number of complications. However, it's a lot cheaper, and in many cases, the exome covers all you need. But that is an important choice that you need to make up front of whether you're willing to restrict yourself to such a small subset of the genome, and whether what you can afford for your experiment overall. Other targeted techniques are very similar to exomes, except they come with additional technical complications, which I won't go into here. Okay, so all right, you've decided what type of variants you're going after. You've selected a sequencing technology. Now it's time to actually generate some data. So the first step is going to make some library preps. That happens in the wet lab. You're going to take some samples, biological samples from individuals, and extract DNA or RNA and then produce complementary DNA that you can actually sequence. The exact techniques is going to vary a lot depending on experimental design, but ultimately what they all have in common is that you end up with fragments of DNA that are typically a few hundred base pair long. You take those fragments of double-stranded DNA, you're going to do some additional preparation, like in vitro processing, doing things like adding adapters, adding barcodes, stuff like that. And that is going to produce, at the end, a library prep that is what you're going to be sequencing. Okay, so that's your library prep. Second part is actually you take your library preps for all your samples, and you're going to deposit them on a flow cell. Here I'm showing Illumina technology because that's what we use the most abroad. Typically a flow cell has multiple lanes. You'll spot your library preps in each lane of the flow cell. At this point you can choose to multiplex, which means that you can put the same library across multiple lanes. You can also put multiple libraries on the same lane. The choice on that has to do with how much you want to spread your risk, because some lanes sometimes will fail, there'll be some kind of problem. So if you spread the library across multiple lanes, you can reduce the risk of not having any usable data. You can put multiple samples together on the same lane to cut down on the cost per sample. But whichever you choose to do, what you end up with after the sequencing process, and Illumina has some really nice videos that explain exactly how their chemistry works. Other manufacturers have equivalent documentation. What you end up with is an enormous pile of short reads that are typically identified by read group. And that's a very important term in our pipeline. The read group comprises the sequence that's produced from one library prep on one lane. So the part of the library prep that I spotted on lane 1 is going to yield one read group's worth of sequence data. If I multiplex, I'll have multiple read groups. I have to demultiplex based on barcodes, etc. Being able to identify read groups accurately allows us to, in the next steps, to clean up some sequencing artifacts, things like that. So it is very important to be able to track your read groups accurately. Okay, so at this point, I have my raw sequence per read group. Typically, people get their sequence in FASTQ format. That's the most common format. Now, the hitch here is that at Broad, we don't use FASTQ as an intermediate. We use another format called an unaligned BAM. I'll talk about that briefly next. But generally speaking, for our YouTube audience, people tend to have their sequence in the FASTQ format. And in both cases, the key information is the same. It's that you have your sequence name, and here in my example record, the first line is going to be my sequence name. There's information in there about the flow cell lane and various technical things that, as an end user, usually you don't have to worry about, but it's good to know that it's there. You'll have your sequence, the actual sequence that was read out by the sequencing machine. There's a third line that's kind of vestigial. I'm not sure what it was ever used for. You can have the read name repeated, but I have not, in many years, seen any FASTQs with anything in that third line. And then the fourth line of your FASTQ record is the base quality score string. This is super important, and I know it looks like gobbledygook, but it's actually quite important. Each of these characters in ASCII code translates into what we call a Q score. That's your quality score. There's one for each base call, and this translates into a number that tells you how confident the sequencing machine was that it read out the correct base. And the way it scales, because it's reported on a log scale, which makes it very easy for us to manipulate those numbers, those quantities in our puny human minds. The way it scales is that if you have a Q10, a value of 10 for your quality score, that tells you that the machine was 90% confident it made the right call. It might seem good, but that's actually pretty bad. That means that 1 in 10 base calls will be bad, which on the scale of billions of base calls is actually quite terrible. If you scale up, you have a Q20, it's 99% confidence. Q30, that's 99.9% confidence. That's where we're getting into good values. But you can see immediately that it's a lot easier to think about numbers between 20 and 30, 40 than having to think about a large number of decimal places. Okay, so this scaling allows us to manipulate these quantities much more intuitively. So that's the point of that. And again, we have one per base. The ASCII code is what allows us to have that one-to-one correspondence.
The semicolons versus the numbers.
So the fact that here I have semicolons versus numbers, I could also have letters. It doesn't matter. It's a long list of characters, and they each arbitrarily have an integer value. So there's no significance to the fact that it's a punctuation versus a number versus a letter. Now, if you're very familiar with ASCII code, you can read that and know what range of values they're in. But honestly, there are tables, lookup tables for that. All right. So that's how our basic raw sequence is encoded. At this point, we have our big pile of reads, and we're going to put that into what we call the GATK best practices pipeline. And that's a set of processing steps that's going to allow us to go from reads to variant calls. We're going to go through 3 main phases of analysis. I'm mostly going to talk about the first 2 because the third is very project specific. The main 2 phases here are the data pre-processing, where we're going to align and do some denoising, some cleaning up of the data. And the second phase is variant discovery itself. The first phase is going to be pretty much the same for all the types of data that we push through, whether we're looking at germline or somatic and the types of variants. But the second phase is what's going to be quite different between the cases. All right, so let's look at that first phase. I have my raw reads. The first thing I have to do is align them to my reference, because I need to know for each read where it sits in the genome so that I know what genomic region it informs me about. So I'm going to read by read, do a global alignment, and find which position, which area of the genome my read fits in. The output of that is going to be a new file in which we've mapped the reads. That means we have some extra information. Here we're going to have a different format to represent that information. It's called a SAM or BAM file for sequence alignment map. BAM is the binary compressed version. So it's compressed to take less space because these files are very large. Can be hundreds of gigabytes for a whole genome. So the BAM file is more compressed, but it's essentially the same content as the SAM file. This is what one read record looks like. Each read is on a separate line. You'll have the read name, which is repeated. We already had that in our FASTQ. You have the read sequence and the quality scores, which we already had in our FASTQ as well. But now we've added additional information. In orange, you see the mapping information that the mapping software has added, and that's things like the position, where it maps in the genome, which chromosome, which starting position. It tells us the cigar string. Cigar is a way of representing the structure of the alignment. It tells us how many positions match, then whether there's gaps or insertions in the alignment. And this is represented in a very succinct, compact way using this this notation where 23M means 23 matches, and so on. We have additional documentation about how to decipher that. I'm not going to go into it here. And also you get information about whether, if you're working with paired end reads, you get some information about the mate, and you also have some mapping quality, which tells you roughly, it's not very well calibrated, but it tells you roughly how well the read mapped. And this is important for distinguishing reads that we've placed confidently in a particular place versus reads that could map elsewhere in the genome, which typically are less reliable. They inform us less, and we want to know that. There's also some flags that refer to various properties of the reads and some metadata that can describe what sample this read belongs to and so on. This information is, like I said, represented in this BAM file, and it summarizes really the position, the quality, and the structure for each of the sequence reads. So going from a FASTQ to a BAM file, it's not just a format conversion. We've also added new information about mapping. I mentioned that at Broad we use an unaligned BAM file as initial input into the pipeline instead of FASTQ. So it's essentially the same, except you wouldn't have that positional information. And the reason we do that, there's no scientific difference, but it's In terms of data management, it allows us to plug in the metadata for all the read groups at an earlier stage, and it makes it less likely that things are going to get lost. Yeah. Okay, so once we have that, we have our map file. What we want to do is do a few processing steps that are going to clean up the data. We need to clean up a few artifacts. The first thing we're going to tackle is duplicates. Duplicates are when you have reads that are duplicates of each other, meaning they were read off of the same DNA fragment. That means we have non-independent measurements of that sequence. And if we counted them all equally, that would inflate our counts when we're counting support for or against a variant allele. And so we want to identify those. For example, here I have a pile of reads aligned to a reference that are duplicates of each other. And we have algorithms to identify that. I can mark them. I don't want to remove them. We don't like to remove data, but we can identify them and tag them so that in downstream steps, tools like the variant caller can safely ignore them. So that we're only going to count that observation once and avoid making calls that are skewed due to this type of technical error. Okay? So that's the first step. Here we have a second step that is optional. It's something that was very useful back in the day when the variant callers were trusting the position, the mapping given by the mapper absolutely. Now our variant callers actually reassemble the reads. One of the first things they do is throw away the mapping information, the local mapping information, and try to remap the reads on a local scope, and that makes it possible to avoid some artifacts. Specifically, this type of artifact is one of the type of artifacts that is caused by the naivete of the mapper. The genome mapper only maps one read at a time, and so because of how it scores the alignment, it can make mistakes, especially towards the end of a read. If I have, for example, here I have a homopolymer. It might be a little difficult for you to see from there, but we have several repetitions of a T base, and that's going to cause some difficulties at the alignment step. If I actually have an indel, here I have a deletion of one base in this data, the mapper is not able to identify that deletion properly, and so it's going to map all the reads with these mismatches towards the ends of the reads. And this is purely because of how the scoring scheme is set up, and because the mapper is only seeing one read at a time. But if we do a realignment step where we look at all the reads together, we can identify that there's actually a one base pair deletion here. And clean up this entire alignment, we can identify a variant, an actual deletion that we wouldn't have seen otherwise, and we avoid calling all these spurious SNPs that are actually artifacts. And so this used to be very important, but because of progress in variant calling algorithms, we no longer need that for our more modern pipelines. Some of the legacy pipelines still use it, which is why I want to make sure to explain what it does and how it works. So that's indel realignment. Base recalibration is another process that is going to clean up some artifacts. The idea here is that I told you about the base qualities, right? The base quality scores. So for each base call, I have a confidence estimate. However, we know that the sequencing machines, when they're calculating those estimates, can make mistakes. And not just random mistakes. Random mistakes would be fine. We can deal with that. But they make systematic mistakes due to some particular technical properties. And one type is, for example, here, based on sequence context. here I'm showing that based on the sequence context, meaning the bases that precede the base call, we can find that there are some systematic errors that are made. For example, here, what this is showing, I want, ideally, if there was no error, this is empirical minus reported quality. We gloss over that in the interest of time, but I'm measuring this systematic error. Ideally, I want everything, all the lines to be lined up on 0. If I have something that's higher, that means that I actually underestimated the quality of my base, and that happens here in this case. Every time when I have an A and then an A, the next base that I call, I tend to be more correct than I expect. And here there's a systematic bias. For some reason, I'm underconfident in the quality of my call. I should be more confident. Conversely, we have an example here with another dinucleotide context covariate, where every time I have this particular, it's not AG, but I'm going to pretend it's AG. Every time I have those two bases, the next base call that I call, I make mistakes, but at a higher rate than random, but somehow I'm not able to integrate that into my confidence calculation. And so that's going to lead to artifacts in the variant calling stage. So we want to clean that up, and we have algorithms that can detect these systematic errors and correct for them. So to be clear, we're not correcting the base call itself, that ship has sailed, but we can correct the confidence score to have a better estimate of how sure we are that this base call is reliable. And this is going to be very important in the variant calling stage, because we're going to use that quality score as one of the things that go into the calculation of the likelihood of the variant. Okay, so here it's quite important to put the data through this base quality recalibration step. All right. One more note, if you're running with RNAseq, there's going to be small differences relative to this pre-processing pipeline. I'm going to use a different mapper to map my RNAseq data. I'm also going to do an extra processing step. And this is just necessary because if you think about what happens when you're sequencing RNAseq, you're not sequencing the original genome sequence by definition. You're sequencing something that has been spliced, and at the splice junctions, you're going to have some reads that overlap, and that originally, when you map it back to the reference, would map across the splice region. You need to have a mapper that understands that, doesn't think that these are just massive deletions, but that they're actually splice junctions. And so there's a little bit of special handling to deal with that. That is also described in our documentation. It's all about handling splice junctions. Okay, but that's the only real difference if you're running RNAseq compared to DNAseq. In either case, once you've run through this pipeline, after base recalibration, what you have is going to be analysis-ready reads in a BAM file that's ready to feed into the variant discovery phase. So again, this first part was all about mapping and dealing with artifacts, dealing with technical errors. In this phase, we're going to look at our lovely clean data, and for each area of the genome, we're going to try to identify what variants might be there. Now here, and think about, this is what the data looks like when it's super clean. In this case, this is an IGV screenshot where I'm showing all these gray bars. Each one is a sequence read. They're aligned to the reference, which is represented here. The top track gives you the amount of coverage, so the amount of sequence that you have at each position. And in this case, we're showing a very clean, probable SNP. So we have one C that's been replaced by a T in about half the reads, so we assume it's probably a heterozygous SNP in this sample. And so that's going to be, for short variants, that's the variance caller's job, is to look at that and determine whether there's enough evidence to say that this is a variant. Now, this is a very clean example. In the wild, the data tends to be a lot more messy. And so that's why we can't just do a naive count, pile up, and decide, okay, it's about 50-50, so we're good. In realistic situations, we often have much more complex situations, and so we have algorithms that are tailored to apply fairly sophisticated statistical models to evaluate whether something like that is a real variant or not. What will be produced? What will be produced by the variant caller in the case of short variants is going to be a VCF. It's called variant call format. It's a text file that will give you, first you'll have a header. We love headers because it allows us to pack in a lot of metadata and information at the top of the file. But then you have the variant information itself that's summarized here below. You'll have the chromosome or contig that your variant has been identified on, the position, the starting position, if it spans multiple positions. In some cases, if you've provided a database of known variants, you can annotate an identifier if the variant has been seen previously. You'll have your reference allele and your alternate allele. So the first line, I have a G to A SNP. The second line, I have a T that doesn't have an alt, so hang on. Not sure why I have a dot there. I'm going to hand wave it back. The third line here is a, oh yeah, because, sorry, coming back, we're all homozygous here. So we're all homozygous reference. We don't have a variant. This is how we represent a fully homozygous reference line. Okay, and my third one here is an indel where I have a deletion of one base. I have a deletion of the T. I'll have some additional information. The qual score is also a confidence score that estimates the quality of my variants. The calibration on this is not very useful for filtering. So for filtering, for determining whether we actually agree with the variant caller, we'll have some more sophisticated annotations that describe the context of the variant. Filtering, if you've applied some filtering, you may have something written in this field which tells you that the variant site was considered but filtered out as a probable artifact. In the info field, you'll usually have a lot more annotations than that. That's where you're going to record things like the depth of coverage that was observed, but also a number of other statistics that we calculate for you that describe the variant more completely and summarize what the data looked like at this site. Then we have format and these additional columns. These are all different samples. So here we have a multi-sample VCF with 3 different samples. This is the genotype information. The names of the annotations are given in this format column. Then here we have the values for various annotation that apply specifically to each sample. The genotype, genotype quality, and various other annotations. So that's how we report short variants. For other type of variants, there's other ways to report it, but this is the very standard way to report short variants. And again, the format specifications can be very useful in deciphering these files if you're not familiar with them. Okay, so obviously I took a shortcut there. I talked about variant calling, then showed you VCF. The big gap in the middle is the variant calling process itself, and that is very different depending on the type of variants you're looking for. For short variants between germline and somatic, we have similar processes, but there's some important differences. And I'm going to go through a comparison of these. I'm also briefly going to describe the pipeline for somatic copy number variation, which is something we've added very recently. So let's look at each of them in turn. For germline, specifically germline short variants, everything we do, we want to do on cohorts. So not a single sample. We want to be able to look at multiple individuals and compare them. Compare the variants they have, what they share, what's unique. So for that reason, so for scientific reasons, and also for technical reasons, we want to have as many relevant samples as possible within a cohort. And so we're going to take multiple samples, and we're going to all pre-process them the way I just described, then feed them in together to this second phase of the pipeline. The first step is still going to be done per sample, where we're going to do initial variant calling with our flagship germline tool, which is called haplotype caller, which many of you may have heard of. And a haplotype caller is going to apply some fairly sophisticated statistical models to identify everything that could possibly be a variant in each sample by itself. And then it's going to write an intermediate file called the GVCF. And then once you have GVCF from multiple samples, you'll put that through joint genotyping and joint filtering. And now I'm going to explain why we're doing this two-step process. Because we want to be able to do as much as possible of the analysis on multiple samples from as early as possible. We don't want to make individual VCFs for each sample and then compare them at the end. For various reasons, this is a great way of underpowering your analysis. What you really want to have is a joint call set that you process together. Like I said, from as early as possible. The traditional way of doing that was to take all of your samples, all of your sequence, and put that through variant calling with haplotype caller at the same time. The problem is that this scales extremely poorly because the compute requirements in terms of performance, memory, and so on get really bad once you get above a few hundred samples. And on top of that, because you have to run haplotype caller on all samples together in this schema, what happens is that, let's say you've accumulated a number of samples, then you joint call them in that one-step process. And let's say 2 months down the road, you get another batch of samples. Well, you have to redo all of the calling. It's going to cost you money and it's going to cost you time. And you're going to hit a wall in terms of the amount of samples you can process together. That's called the N+1 problem, when you want to add one more sample and you have to redo everything. It's horrible. And so the GATK development group was tasked with developing a solution to that didn't just involve throwing more power at the problem. Specifically, that was to enable the joint calling of the EXAC project. And what the group came up with is this really elegant solution of decoupling that initial variant calling, which you can now do per sample with haplotype caller, from the second step, which is where you genotype the samples together as a population, as a cohort. And you can do that with the second tool that runs really fast and that scales linearly with the number of samples. And so by decoupling this process, we've made it possible to scale up to fairly ridiculous numbers of samples. I believe we're looking at 120,000 exomes, and we have a call set of 20,000 whole genomes that recently completed. So that's a huge amount of data, and that was made possible by this workflow specifically. So that's a very specific to the germline SNP and indel workflow. That's kind of the highlight. If you want to know more about how haplotype caller works, how the joint genotyping calling works, we have a lot of documentation on the GATK website that explains that in more detail. Now, to briefly talk about the somatic SNPs and indels, here we have a very different paradigm because here we're mainly comparing a tumor and a normal sample from the same person. And we're trying to use that comparison to identify specifically what is purely somatic versus what was just part of the germline variant load of that person. The challenge here is that there's multiple challenges. One of them is that there's the purity of the samples that we get is variable, and we tend to have contamination from tumor in your normal sample, you have normal in your tumor sample. Within a tumor, you have multiple cell lines that are going to diverge and have different. The result is that your allele frequencies are going to be very different for the sites of interest. Overall, what that leads to is that whereas in the germline context, we have a really good ratio of signal to noise, in the somatic context, it's a much closer call, and so you have to have algorithms that are specifically tailored to that. And so for that, we have a variant caller called MUTECT, the original version, MUTECT1. There's a new version that's called MUTECT2 that calls indels in addition to SNVs, and that's modeled after the haplotype caller, but tailored specifically to address the challenges modeling somatic variation. So you can see that those are very different workflows. Somatic CNV is even more different because there, what you're looking for, when you're looking for copy number variation, what you're looking for is differences in the amount of coverage that you have over a genomic region. This explains how it works in a nutshell for exomes, where you're going to count the amount of coverage you have. You're going to look proportionally overall how much that amounts to. You're going to have some more pre-processing to remove the noise that's inherent in the exome experimental design. And what that will lead to is the ability to identify if this is overall the normal amount of coverage that you have over all your targets, you'll be able to identify regions where you have an amplification or deletion of a subset of the segments. So this is a very new method. The method has been around for a while, but it's been re-implemented recently in GATK, specifically in GATK4, and that's a new feature that's rolling out now. And so that's a kind of newer pipeline that we're able to propose. Okay, finally, the third phase is much more project dependent, so I'm not going to go into detail. But it involves additional filtering and evaluations of your call set. Okay, so that covers the scientific aspects of what happens to the data, what needs to happen in order to identify these types of variants. We have a lot more documentation on these on the GATK website that I encourage you to check out. The software, very briefly, IGV is a very, very useful genomics viewer that was developed originally here at Broad. And that's what we use for actually visualizing the data. And you can use a lot of different data types and integrate them. In terms of genome mappers, we use BWA for DNA, STAR for RNAseq. They have some important operational differences. And again, we don't develop them, we don't support them, but they are what we use in the pipelines. Picard is a toolkit of processing tools, mainly aimed at BAM manipulating and quality control, very important quality control. And we do support that on the GATK forum. So if you have problems with it, we can answer questions. GATK is what I'm most closely associated with. So it's the toolkit that comprises the different variant callers that I've talked about today, plus a number of additional utilities and some of the pre-processing utilities. I want to mention that in terms of development roadmap, we're on the cusp of a really big change in GATK. Our engineers have spent the past year completely re-implementing, rewriting the engine to be much more scalable, to be much faster, and offer a lot of new features that we weren't able to offer previously. So there's a lot of change on that front. More functionality. In the near future, we'll also be able to package the Picard tools as part of GATK, so you'll be able to have all of those tools available in one package. Now, the pipelines in the last 5.5 minutes that I have, I want to give you a summary of the pipelines that we run here at Broad and mention especially some of the more recent developments that are going to make a difference to probably most everybody in this room and a lot of people outside of it as well. The legacy, I would say, for a long time, all our pipelines were run on-site on the Broad clusters. Well, now in Alston, but Broad clusters. And the way it was implemented was that we had a common pre-processing pipeline for all of the samples. And you might have heard this referred to as the Picard pipeline. That does all the base calling, mapping, and processing steps. And it's set up to process things in a very high throughput way, including managing when you have multiple read groups per sample and so on. So that's typically done for you. It's a Picard pipeline. Then once you get your analysis ready BAMs, you go into either the germline variant calling pipeline, which is still part of the Picard pipeline, this optional bit. Or, depending on what you're looking for, you go into the cancer analysis pipeline. This is not pre-run as a service. It's something that you would do yourself as a researcher using what's called Firehose, which is a local pipelining, a DIY pipelining system that's kind of self-service system. And so you see that right now, we're still, everything that's exome is still processed through this, as well as the cancer stuff. However, the big changes, the big improvement that's been added over the past year is that now we also have some of our pipelines running on Google Cloud. The Picard, what corresponds to the Picard pipeline, which is now called Genomes on the Cloud, runs on Google Cloud, and so the pipeline operations group within DSD, within our platform, will run all the genomes that get sequenced now for germline calling get run on Google Cloud. And we share the scripts that are used to do that on a GitHub repo, if anybody's interested in checking that out. So that's Genomes on the Cloud. That's run as a service. There's also same cloud, different but related system, something you might have heard of called FireCloud and or Workbench. FireCloud is available now, and it's led by Gatti Getz's group, and it's a cloud platform that allows you to do self-service cancer analysis research. It's basically Firehose in the cloud, hence the name. The underlying infrastructure was developed, is still being improved in the DSD Data Sciences Platform group. And the next iteration of that is going to be what we call Workbench, which is something we're going to use throughout the Broad and also for external users, make available to external users. And the idea is to provide this self-service pipelining system on cloud, which makes it easier to share data across teams. It also makes it easier to not have to deal with queues. It just depends Like you have a building project and it's up to you to start up some machines. And it's very easy without having an informatics background. That's a really important point I want to make. So this allows you to run these sophisticated pipelines and use the preset pipeline scripts yourself without having to reinvent the wheel of re-implementing all GATK best practices. And it allows you also, if you write your own pipeline, to share that with collaborators or include that as part of a publication. If you publish a paper that describes an analysis, you can share that on this system and make it available to the world. So in terms of reproducibility, it really empowers us to make a big leap forward. That's very exciting for us. An additional note is that in the near future, when you get samples sequenced and analyzed through the Genomics platform and through DSP, the results will be delivered through Workbench, this cloud-based platform. And the idea is really to empower people to really get involved with their own data, but making it easy at the same time. So that's the idea. One more note is you can write your own pipelines yourself if you want to put together an analysis. Part of building this new infrastructure, we also developed a new pipelining language that's really easy to use. And I say this as a former wet lab microbiologist. I worked for 10 years in a environmental microbiology lab, so I don't have a formal programming experience. I can write WDLs. And if I can, so can you. We have a website and a lot of documentation that help you to write your own pipelines and be able to implement or just run things that other people have written. And
is WDL not a domain-specific language?
It is a domain-specific language that is human-friendly. That's the key. That's the ad line anyway. And that's my presentation. Thank you very much for sitting through all of that. I'd be happy